Good afternoon, and welcome to Ask the Doc. This is the live stream series where we invite our audience to ask questions of our experts in Alzheimer's disease and dementia. My name is Dr. Megan Whitbrocht. I'm the Associate Director of Education here at the University of California, Irvine Institute for Memory Impairments and Neurological Disorders. UCI Mind is part of a national network of Alzheimer's disease research centers that are designated by the National Institute on Aging, which is part of the National Institutes of Health. Today, we're joined by Dr. Paul Chadarevian. Dr. Chadarevian is a postdoctoral researcher in the Matthew Blurton Jones Lab here at UCI Mind. He completed his bachelor's in economics and mathematics from UC San Diego before pursuing his master's degree in stem cell biology and regenerative medicine at the University of Southern California. During his PhD studies at UCI, Dr. Chadarevian investigated the therapeutic potential for microglia replacement in animal models of neurodegeneration and was awarded the Carl Kottman Young Scholar Award at UCI Mind, and has a number of articles in high-impact journals. His current work builds off of these efforts to engineer human microglia to deliver therapeutic peptides throughout the central nervous system and identify alternative and less invasive routes for microglial transplantation. Welcome, Dr. Chadarevian. Thank you for joining us. And welcome to our viewers. If you're attending live on Facebook or YouTube, you can participate in the Q&A with Dr. Chadarevian by typing questions in the comment box. Remember that anything you put on the internet lives there forever. All right, Dr. Chadarevian, let's get started. Hi, welcome.
How are you?
I'm doing well. We're really excited to have you on.
Great. It's such a pleasure to be here with everyone.
So can we start with a question that I like to ask our guests? Why did you decide to pursue a career in neuroscience?
Yeah, it's an interesting question, especially given sort of my academic background. I think, like most students early on who catch the science bug, we first think about pursuing a career in medicine. And when I started out in my undergrad and I was volunteering at a number of hospitals, I volunteered at the Huntington's disease clinic, and that was really the first time that I saw the devastating impact neurodegeneration can have on patients as well as their families, their caregivers. And that really sort of inspired me to pursue research in a different way. And that's why I went and pursued my master's. I worked with Dr. Qilong Ying. It was a really interesting and a great experience, but I felt drawn more to translational research other than understanding really the basic science of it all. And so working here with Dr. Matthew Blurton Jones, his amazing team, and I really have to credit his team for really inspiring me, some of the great work done by his previous students, as well as senior scientists like Dr. Hike. That work really sort of caught my attention, and it sort of came naturally to me that I really wanted to sort of make my mark and contribute to some of the great ideas that Matt had cooking in his lab. And it's been, history ever since.
Excellent. Okay, so last month we spoke to Dr. Lindsay Hofsfield about the important role that the immune system plays in protecting the brain. Can you just review with us what the immune cells of the brain are and what role each of them play?
Absolutely. So the predominant and innate immune cell of the brain are microglia, which are the cell types that we study. These are extremely dynamic cells. They're involved in immune surveillance, and they're sort of the first line of defense to infection, to unwanted protein aggregates within the brain. They also play an important role in brain development and in maintenance. They sort of regulate homeostasis as well. But there are also these perivascular macrophages that live near the blood vessels, and they help facilitate and sort of keep the integrity of the blood-brain barrier. And lastly, and this is some work really pioneered by Yoni Kipnis in his lab, we have these lymphatic connections that sort of join the brain through the meninges, where here, as well as within the vascular, other peripheral immune cells can sort of make their way and interact with microglia within the brain. For example, T cells seem to play a very important role, and they sort of act both as a protector, potentially also as a contributor to neuroinflammation. And we really want to understand how microglia can impact these cells within the periphery, how the periphery can impact the microglia deep within the brain. And hopefully by understanding these relationships, we can better address sort of the unmet needs that we find within people with AD.
Very good. Can we focus a little bit on microglia? Because I know that's the bulk of sort of the research that you do. How are microglia implicated in neurodegenerative diseases like Alzheimer's disease?
It's a really interesting cell type because it seems to play both a protective as well as a detrimental role, right? So, for example, they can engulf and clear amyloid within the brain. As we start to accumulate higher levels of soluble amyloid, it can sort of compact it and wall it off from the rest of the brain. But over time, they can also become chronically activated. They can sort of engulf way too much of these proteins, and they can express a very irritated and pro-inflammatory state. And those pro-inflammatory cytokines and chemokines can wreak a little bit of havoc as well. Additionally, it's important to note that a high number of AD risk genes are also found predominantly expressed by microglia within the brain. And so work done by ourselves as well as others have tried to investigate how these mutations and how these genes impact or regulate microglia function within disease. And so, for example, TREM2, there's a number of TREM2 mutations which we've identified exhibit sort of a loss of function on TREM2. And what this does is it actually alters the microglia state and makes them less effective at addressing amyloid depositions within AD brains. On the other hand, there are other mutations like in PLCG2, which have been shown to have a protective quality. They can sort of enhance the immune ability of these cells to address what's going on within the brain within AD.
So I want to investigate that a little further and just understand what you're saying. So you're saying that some of these Alzheimer's disease risk genes that we've identified 60, 80, many dozens of these genes. A lot of them are related to the function of microglia. So it leads us to believe maybe that microglia may be highly implicated in Alzheimer's disease just based on the association with these risk genes.
That's correct. However, it's important to note that it's not only related to microglia. So for example, the most one of the well-studied risk variants are in ApoE, in ApoE type called ApoE04, which is most abundantly expressed by astrocytes within the brain. And so microglia play an important role when it comes to how ApoE is contributing to the onset to the progression of AD, but it is also other cell types that could play a role. The work that we typically do is really understanding those mutations that are predominantly expressed by microglia, which aren't the only mutations that arise, but for example, like the TREM2R47H or the PLCG2P522R, these are mutations that seem to target microglia specifically. And so by understanding the role that they play within microglia, first in a dish and then within animals, we can better understand how they're contributing to the disease pathology and better develop drugs or other therapeutic, strategies to sort of compensate for a loss of function or to help sort of promote a protective quality that microglia could have.
So in an inflammatory state, are microglia good for the brain or bad for the brain?
Yeah, they absolutely play an important role. And one of the unfortunate sort of side effects of the ability of microglia to engulf so much amyloid and sort of act as our first line of defense, that they could become chronically activated. And in that case, they can actually become oversaturated with these lipids, with debris, and they can induce significant oxidative stress on the surrounding neurons and lead to synaptic damage. And it can also impact the states of those other cell populations within the brain, for example, other glia. So other than microglia, we have astrocytes, we have oligodendrocytes, and they play very specific and unique roles, but microglia sort of communicates with them. And when they become chronically activated, they sort of lose the ability to tamp down or regulate the response of these other cell types as well, which can further contribute to neuronal loss and other damages as well. So for example, oligodendrocytes, they play a really important role in myelination, which sort of helps protect and maintain the quality of neurons. And so as microglia become chronically activated, they can sort of lose the ability to tamp down or regulate that oligodendrocyte response, which can then impair their ability to myelinate, at appropriate levels. And also, it also interferes with the ability of microglia to deal with the myelination as well.
I see. And then what is the phenotype of mice who don't have microglia?
It's a great question. It's a really interesting model. It's one that took a little bit of time and development. It was developed by doctors Claire Priddens and David Hume. where they looked at the genetic structure of CSF1R, which is a receptor that is very important to microglia viability within the brain. And they identify that there is this enhancer, shorthand FIRE, an FMS intronic regulatory element, which, if it's deleted, allows for the majority of the peripheral macrophage populations outside of the brain to develop without impairing the general health of these mice. However, within the brain, the mice develop without any microglia. And so what happens is we've done a number of studies now using this model. Not only does it allow for microglia engraftment, of course, transplanting into that open niche, but we've also examined these mice as they develop over time. And what we found is that in the absence of microglia, these brains begin to develop a number of hallmark pathologies associated with CSF1R related leukoencephalopathy. So they start to develop calcifications, they develop reactive astrogliosis, they develop spheroids, which are swellings within neurons, they develop white matter abnormalities as well. And so what we've been able to do using this mouse is sort of mimic the extreme case of that CSF1R related leukoencephalopathy, where patients can develop with homozygous mutations lacking any CSF1R, lacking any microglia in the brain. And so what we found is that if we transplant healthy microglia early, it can prevent these diverse neuropathologies from forming. But if we take older mice that have already started to exhibit these pathologies, we can take a patient line, we can CRISPR correct it, we can correct the mutations within CSF1R, and then we can transplant this into that mouse, and it begins to actually reverse these pathologies that have already sort of taken hold within the brain. And so it's been a really interesting model for us to use to understand the therapeutic potential of iPSC-derived microglia, but it's also been a very useful tool as well that allows us to really study microglial engraftment without mouse microglia sort of impairing our results or sort of neighboring the human cells as well.
Okay, you're going to have to break that down for me, certainly. Can we start with CRISPR? You use the term CRISPR. I hear CRISPR a lot. What is CRISPR?
Yes, it's a very hot topic and a very important one as well. CRISPR is an acronym. It stands for Clustered Regulatory or Regularly Interspaced Short Palindromic Repeats. Essentially, it describes the observation. And it's a gene editing tool. CRISPR and Cas9 work together and allows scientists to precisely modify DNA, which allows us to either correct a mutation if it's something we want to see or understand the effect of. It allows us to induce a mutation, but it also allows us to insert large, complex sequences of DNA as well. So we can generate reporter molecules. We can understand where certain genes are expressed. We can make them inducible. And these two components, the CRISPR and the Cas9, were discovered in 2012 by doctors Duna and Charpentier, who went on to win the Nobel Prize in Chemistry in 2020. And they identified in prokaryotes that there is sort of this immune response to viral infection. And what they found was that within the genome, there were these repeats, these short palindromic repeats that contain bits of coding sequence that relates to this virus or that virus, and it's sort of a library. And what they discovered was, as a viral infection takes place in the prokaryotic cell, that little bit of the genomic information is then produces an RNA, which can then bind to the incoming foreign genetic information of the virus. And by binding to it, this Cas9 protein can then come in and cut it up, essentially preventing the virus from entering its reproductive cycle and integrating into the host. So this observation in bacteria has now moved forward in time and allowed us to make CRISPRed mice, where we're able to engineer the genome of mice to exhibit specific mutations or specific reporters, right? It's also allowed us, like the work I described previously, we can CRISPR correct cells that carry a mutation to transplant them and see if it's able to reverse pathology. Or more so, if there's a very rare disease that we're unable to get a biological sample of, we can induce a mutation in cells to understand and really explore how it interacts with, let's say, microglia or other cell types within a dish, and then eventually move that into mice as well. So it's a powerful tool, definitely something that is being used throughout research, and it's proven actually in some cases to be effective within clinical translation as well.
What diseases has it been shown to be effective?
It's proven to be, it's been explored in many diseases, not only neurodegeneration, but of course those in the periphery as well. So one, for example, is sickle cell disease, which we know the single mutation that causes it, and they've explored in conjunction with bone marrow transplant, correcting the mutation that gives rise to those abnormal blood cells within the periphery. It's also been used, I'm aware, in a rare condition of blindness, this LCA10, in order to inject these CRISPR-Cas9 directly into the eye and to correct the mutation. But it's also being used to engineer immune cells for immunotherapy. So specifically in the treatment of B-cell malignancies, the development of CAR T-cell therapies have really proven to be quite effective in treating these specific cancers, where we're able to give a T-cell the ability to recognize or preferentially recognize specific types of cancer cells. And that really enhances their ability to target them within the periphery and to clear them up. But it's also been considered in the use of HIV as well, to knock out the receptor that the HIV virus uses to actually integrate its genome into the host and cause the disease to spread. And of course, in academic settings, it's been used to treat a number of neurodegenerative diseases, a number of peripheral diseases as well in the organs, for example, in diabetes, other retinal issues as well. So it's quite a vast sort of field, and we really hope to sort of make our mark on the neurodegenerative side of it.
Yeah, let's talk about its potential role in fighting neurodegenerative diseases. Can you expand on that a bit?
Sure. So those CAR T-cell approaches I mentioned earlier, some groups have considered, how can we engineer these cells to actually deal with diseases within the brain? For example, glioblastoma, this type of cancer that develops quite rapidly and aggressively within the brain. But they're also being deployed by other labs to consider their ability to target amyloid aggregations, tau, another type of pathology that develops within AD. Other groups are trying to use CRISPR-Cas9 systems to actually edit or alter the genome of neurons within the brain as well in order to sort of affect these protein pathways that are resulting in disease. But we found it to be quite useful within the work that we do, which is really focused on engineering iPSC-derived microglia and delivering them in a significant proportion of the brain in order to allow for either the prevention or the rescue of different pathologies. And so this process is called microglia replacement. A number of groups have used bone marrow as well as a number of small molecules to help get these peripheral macrophage populations within the brain. But we found it to be quite useful in neurodegenerative models, such as in AD models, to actually replace diseased microglia with healthy human microglia. And so we're now studying that in a number of diseases, and we hope to really get some of that data out as quick as possible.
Yeah, so can you just break it down in simple terms how you do this microglial replacement?
Sure. The first piece of it is that, as I mentioned earlier, CSF1R, this receptor, is really important to microglia homeostasis and viability within the brain. So it allows microglia to stay really happy and allows them to do their job. And there's a number of compounds, this is work also done by Dr. Kim Green at UCI, that if you were to give it to a mouse, it would actually inhibit that receptor from doing its job. And it actually results in the depletion of microglia. It eliminates microglia from within the brain. And so we asked ourselves, could we engineer microglia in such a way where we retain the normal function of the CSF1R, that receptor, it's able to still stay alive, to proliferate and do its job. But for whatever reason, we will prevent that small molecule from binding to the inhibitor. So essentially giving it the ability to have an inhibitor-resistant receptor. And so what we found by studying the crystal structure of how the receptor and that small molecule binds together was that there was a single amino acid that we hypothesized if we could enlarge it, make it slightly bigger, it would be large enough to push out that molecule, but not impair the signaling of the receptor into the cell. And so this mutation, G795A, we found that if we knock in this mutation to healthy microglia, we transplant it into a mouse model, we're able to then feed the mouse these inhibitors. As the mouse microglia begin to deplete, the human cells, which are resistant, take over, they proliferate, and they begin to engraft throughout the brain. And what's really exciting is that we found these cells will also make their way down into the spinal cord as well, and that even after ceasing treatment, when you stop giving the mouse these drugs, the new cells, the healthy cells, don't go backwards. They don't retract from their newly engrafted areas. So we're essentially able to replace the endogenous microglia with our CRISPR engineered microglia. And that really sort of opens up a lot of avenues for us, which we've began to explore within our lab.
Very good. So what I'm understanding is that these new microglia aren't the chronically stressed microglia that we're now depleting, that we're in there trying to gobble up all of the protein aggregates, that these new microglia are fresh, they're ready to get to work, they're not sort of chronically inflamed. Or
yeah, that's definitely one important way of deploying these cells. And as we discussed earlier, there are these mutations that we can find, for example, in AD that are predominantly expressed by microglia. And so one thing we're exploring now is not only replacing microglia with young, healthy microglia, but if we were to correct those mutations, and now we put it into a brain where they previously had these dysfunctional, chronically activated microglia, could that begin to either slow the disease or reverse the disease? And then we're also sort of taking a second approach where we're not only giving them healthy, happy microglia, but could we engineer microglia in a way to have an added sort of bonus feature? Could they produce an enzyme or a therapy which we've previously found is really limiting to get into the brain if we deliver, let's say, from the periphery, from an IV or from the blood? And so we've been doing a number of studies now in Alzheimer's disease models in mice where that seems to be the case, that we're able to engineer microglia, not only correct mutations, make them resistant to these drugs and deploy them in the brain, but we can actually knock in some type of therapeutic protein, and that these cells will not only produce that protein, but we can engineer them in such a way to do so at the sites of pathology and to really do it in a site-specific manner, which has proven to be quite effective so far within our hands.
Excellent. And you're doing these in mouse models right now?
We're doing them now in mouse models, and that's sort of our bread and butter within the lab. Of course, we do some preliminary work in vitro where we study these cells in a dish, understand how they function, and we're really excited to really see how these cells, once deployed in sort of the more complex environment of the brain, where we're no longer only looking at microglia, we're looking at how the neurons and how the other cell populations are reacting, how is the inflammation changing, how these sort of designer cells could then implement a therapeutic efficacy in a way that either prevents or reverses the pathology once it's been set in.
Science is so cool. Your research is so cool.
We're really privileged. We've made, Dr. Blurton-Jones, Matt, has made a considerable number of advancements in the field, and the work I do now really stands on the shoulders of everyone who's came before me. And so our ability to make these cells, first and foremost, our ability to figure out what type of mouse do we need in order to transplant them and sort of study them in vivo, our ability to develop really robust ways to clear out the dead microglia, all these things were other discoveries, including the work we discussed earlier about identifying CRISPR in these prokaryotes and these bacteria. All of that sort of feeds together in a funnel, which has now sort of given us this next level, this next thing that we can use to bring all of those previous findings together. So it's really exciting. And again, it's having previously been interested in medicine, I don't feel I've really strayed away from that. I'm still patient focused. I really would like to figure out a way to develop these therapies or these treatments that can really benefit the lived experience of these individuals. And it really has to sort of take place here at this junction between basic science, where we sort of figure out how these cells work, we figure out how the disease progresses, and then sort of the next step, which is, well, how do we get them then into a clinic?
Yeah, absolutely. Well said. Okay, kind of along those lines, are there any concerns, ethical, other concerns about this technology that your lab or maybe other clinical scientists have to consider?
Sure. There's a number of limitations and, of course, concerns as well. For example, immune rejection is one. Just as when we do a liver transplant with an individual or we do a bone marrow transplant, we need to make sure that the donor and the recipient match to a certain extent to allow for that organ or to allow for that bone marrow to engraft. Perhaps it has to be in conjunction with certain immune suppressants as well. There's also certain limitations right now on how we get these cells into the brain. So far, most of our work has been by directly injecting them into the brain, into actually the cortex. And so we really want to find less invasive routes that we can use to get these cells into the CNS. And we're really lucky so far that we've identified a couple, and we're really piloting those out now in mice to sort of better understand the limitations of those. Ethically speaking, there are, of course, limitations in the use of gene editing in general, whether it's CRISPR, zinc finger nucleases, and there's been a lot of consideration on at what stage could these technologies be used. Is it acceptable? Is it reasonable? Is it ethical to do this at the to do this on gametes, to do this on eggs, on sperm, in order to correct mutations an individual already carries? Is it ethical to do it on a embryo after those two have already joined together? And there's been a lot of discussion on really understanding what are the impacts of these things and what are sort of the secondary consequences of them. Fortunately, the work that we're doing in the lab, really understanding how microglia replacement, how CRISPR engineering these cells can address neuropathologies is really focused on more adult individuals once they've actually understood and sort of been recognized as carrying these diseases. For example, someone with Alzheimer's isn't necessarily born with Alzheimer's, but they will develop it over the course of their life. And we're really trying to focus on not only preventing disease if we get these cells in early on, but how do we really address the concerns of patients who already have these neuropathologies? Could we reverse them? Could we get them in at a certain state in a safe way, with the right molecules or with the right sort of treatment plan to limit the amount of secondary pathologies or to limit the amount of inflammation, to cause the least amount of damage, and to really allow these cells to sort of do their job, which is really acting as sort of the innate homeostatic regulator of the brain. And so we hope some of those limitations could be addressed in the work that we do. We know that there's other collaborators as well, sort of addressing them in other diseases. And so it's going to be really interesting to see how the regulatory landscape, as well as how the sort of development of these technologies and these therapies translate from what we do within the lab to eventually the bedside, where we can actually deploy them within patients.
Yeah, excellent. So we have a question from our audience. How soon do you expect to see this therapy be moved to a human model?
That's a great question. And it's a question, unfortunately, we don't know the specific answer to, because it is somewhat of an uncharted territory. So developing these therapies, proving their clinical efficacy, getting them into the clinic all requires sort of this dance with the FDA as well, and getting, you know, approval that first and foremost that these cells aren't going to cause other problems, and that once they get into the brain, we're able to then control their ability to be effective, and that they don't eventually go bad. And so, you know, that can take anywhere from 2 to 5 years. And so we hope with other partners that we're working with and other industry leaders that that pathway, especially for some of the really rare diseases where patients have an absence of microglia, perhaps we can, you know, really address those concerns quite rapidly. So if you ask me for my estimation, I would, you know, hope that there is sort of a pathway within the next, you know, 2 years to really start examining these cells within humans and see if they're able to really impact the disease in a meaningful way that we hope they do.
Excellent. Okay, can you finish up by telling us what you're working on right now? You've touched on it a few times, but summarize it, and then tell us where we can learn more about your research.
Sure. We're currently wrapping up a really interesting study where we engineered microglia to deliver this amyloid protease, this enzyme that cuts up amyloid called neprilysin. And we found that by engineering cells in a inducible manner, that they migrate to the pathology, they produce this enzyme, it lowers amyloid levels, it lowers levels of neuroinflammation, but it also increases, or rather prevents, the degradation of synaptic markers. It helps preserve the neurons as well. So it's really exciting. So we hope to wrap that up very quickly soon. We're also exploring the deployment of microglia in other diseases. So there's a really talented graduate student in our lab who's working on a number of lysosomal storage diseases. We think it'd be quite effective there as well. And we're also working on other neurodegenerative diseases too, like frontal temporal dementia. And some of the latter work sort of that we're trying to really get our hands around now is really adjusting some of those limitations we discussed earlier. You know, are we able to, you know, do these microglia transplantations with in an immune compatible way? And so we're developing some new mouse models that really allow us to investigate that. And that work can take anywhere from, you know, again, 2 to 6, 7 years. You know, it's a big project, but hopefully, you know, we make that journey and we're able to share it with everyone in the field. You can learn more about the work that we do in the lab at the website for the Blurt & Jones lab shown here on the screen. We're also regularly publishing our papers and making them open source. So individuals who are really interested in learning more about the techniques and how we use this can find them on PubMed. And wherever possible, I mean, we're happy to share this information and really educate those who are interested in the field.
Thank you, Dr. Chadarevian. I really appreciate you joining us. This was a fascinating talk, and we're going to have you come back and talk a little bit more about this.
It'd be a privilege and an honor. I think you guys are doing really great work, and thanks to the whole team at Ask the Doc and all the great work at UCI Mind. I mean, without the support of UCI Mind, we wouldn't be able to do the work that we do. So thank you so much. I'd love to be back, and thank you again.
Yes, absolutely. And thank you to our audience. Thanks for joining us. This was a great discussion. We'll be back next month with Dr. Michelle McDonald. She's going to talk to us a little bit about the differences between normal aging and aging with dementia, understanding symptoms. And it'll be a really good conversation too, just something a little bit different. If you want to learn more about UCI Mind, visit our website at mind.uci.edu. And then if you want to learn more about our media platform, Mindcast, just /Mindcast. To get involved with research, you should join our registry at c2c.uci.edu. Until next time, take care, everyone. Bye-bye.